بسم الله الرحمن الرحيم. النهارده هنتكلم على الجينيتك كود وريجوليشن اوف بروتين سينثيز. الاوتلاين جينيتك كود، regulation of protein synthesis, regulation of gene expression in prokaryote, positive and negative regulation, and genetic the ribosome from bacteria recognize AUG as start codon with the aid of special sequence called Delgarno sequence. As we see in this figure, this is the Shino Delgarno sequence is a messenger RNA which is complementary with the ribosomal RNA to help the start code. In this figure, this is the initiation complex in prokaryote and this is the initiation complex in eukaryote. The initiation complex in prokaryote, which is the ribosome as we see in the 70S ribosomal subunit, AUG on mRNA, which was the red color, and code with the methionine, which carried on tRNA. Also see in the 80S and EU. This is the table 3 which code for different amino acid. Okay, mistranslation. What is mean by mistranslation? Mistranslation mean the codon may be read improperly and the wrong amino acid inserted. Amino acid whose codon differ by only single base, for example, as phenylalanine UUU and leucine UUA, are most likely to be mistranslated because only 2 of 3 bases are involved in codon-anticodon recognition. In the normal cell, this rare error occurs in only a small number of all protein molecules and hence no determined effect. What is meant by overlapping gene? Overlapping gene means reading of the same nucleotide sequence into different reading frame beginning at different site, and this occur mostly in the virus. Regulation of protein synthesis. Regulation of protein synthesis is induction and repression. The amount of enzyme in the cell can be controlled by increasing, what is mean induction, or decreasing is repression, the amount of messenger RNA that encodes the enzyme. This transcription regulation involves interaction between regulatory protein that bind to DNA to small molecule is called effector protein. For one type of transcription regulation, the regulatory protein is called a repressor protein, and its function by inhibiting This is to demonstrate this is an example of the regulation of positive and negative regulation for gene expression in prokaryote. The regulation of transcription depend on 2 interaction or 2 types of interaction, protein and DNA. The promoter is the site of the DNA that participated in this interaction. And the protein bind to this site or RNA polymerase. When RNA polymerase bind to promoter site, the transcription can start. This is the positive regulation and this is the negative regulation. The activator protein will bind to near to promoter site, so the transcription of the gene, which is the green color, is okay. In negative regulation, the protein called the repressor protein, when bind with the operator, no transcription to occur. Okay, when operator repressor not bind with the operator, the transcription is okay, so it is called negative. Okay, and this example of lactose operon, which it consists of different gene with structure gene ZYE, which translated onto protein or enzyme on messenger RNA, which translated into protein, which is the enzyme beta-galactosidase, permease, and transstylase. And this is the promoter site with the yellow color and operator site with the purple color. Every gene must have a promoter site or it cannot be transcribed. The DNA segment near the promoter site as a binding site called activator protein, and repressor protein. In bacteria, most binding site for repressor protein are called operator. So what is mean by promoter is the DNA which bind with the activator protein. What is mean by operator is the DNA which bind with the protein called the repressor. What is mean by positive and negative regulation? Positive regulation, for some gene, an activator protein must bind to its target DNA as necessary prerequisite for transcription to occur. So it is called positive. Why? Because the bound protein is required for transcription. Negative regulation. For other gene, a repressor protein must be prevented from binding to target site as necessary prerequisite to transcription to begin. Why it is called negative? Because the absence of the bound repressor allows the transcription to begin. This regulatory protein and their binding site on DNA constitute what is called genetic switch. What is the importance of genetic switch? Control the efficient change in gene expression that occur in response to environmental conditions. So, what is mean by genetic switch is the regulatory protein and their binding site on DNA. What is the importance of genetic switch? Control the efficient change in gene expression that occur in response to environmental conditions. Give an example to explain what is how the gene is expressed or how a genetic switch work. Example of lactose structure gene. The lactose operon consists of 3 structure genes, Z, Y, A gene, which when expressed to messenger RNA and translated into protein give 3 enzymes, which is permease enzyme, beta-galactosidase enzyme, and transstylase enzyme, which is important in metabolism of lactose sugar. How lactose operon switch work? In absence of inducer, which is the lactose repressor bind to operator and prevent the transcription of lactose operon, so block RNA polymerase and the structure gene of lactose operon ZYA gene are repressed. In contrast, when inducer present, it bind to allosteric site of and the enzyme beta-galactosidase, permease, and transstylase cell. As we should see in this figure, A, no lactose present in the above, and B is the lactose present. When no lactose present, the repressor protein will bind with the operator, so the structure gene ZYA not work, no expressed. When lactose present in the P site, the inducer, which is the lactose, which when present, will bind with the repressor, allowing the no binding of repressor with the operator. So when the repressor not bind with the operator, so the structure gene ZYA gene are expressed, give messenger RNA, and the messenger RNA are translated into protein, okay, and give these three enzyme, which is the beta-galactosidase, permease, and transacetylase, which is used for metabolism of sugar. This is the example to illustrate how lactose operon work, to explain what is the gene on or gene off. Okay, gene on and gene off or switch, genetic switch, like بالظبط كده. انا مثلا دلوقتي في نور ربنا موجود، فمش بقيد نور، فبيحصل جين اوف. طيب لو مفيش نور ربنا موجود، يعني بقينا بالليل، بقوم عامله ايضا نور، فبعمل جين اوف، جين اون، وبالتالي الخليه عندها المفاتيح الجينيه دي بتعمل اون او اوف حسب مين؟ حسب الانفايرمنتال كونديشن. بمعنى ايه؟ بمعنى مثلا هو دخل لها لاكتوز، هي عاوزه تعمل له ميتابوليزم، يبقى هتعمل جين اون. طب لو ما دخلهاش لاكتوز، هل هتعمل اكسبريشن للجين؟ لا، لان هي مش محتاجه ان هي تعمل ايه؟ ميتابوليزم للشوجر ده. وبالتالي ده مثال توضيحي بيقول لك عشان اوضح لكم ازاي الخليه ممكن تعمل اكسبريشن للجين. ويعني ايه اكسبريشن للجين؟ يعني تديني فانكشنال جين، يعني تديني ماسنجر ار ان اي. الماسنجر ار ان اي يترجم الى بروتين اللي هما الانزيم اللي هو يعمل لي ميتابوليزم للشوجر. وشكرا.